a good morning or afternoon, and thank you for your patience there. Thank you for joining us today. I'm Jan Reichert, Executive Director of the Antibody Society. This webinar is one in a series that's designed to inform and educate our members, as well as the broader scientific community, about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIRR community. Our speaker today, Dr. Vadim Nazarov, is an active member of the AIRR community. Today, he's going to tell us about easy, fast, and practical AIRR analysis. In particular, he will discuss the exploration of single-cell and bulk immune repertoire data in R using Immunarch with application to immunotherapy. The webinar has several parts. After concluding the first part, we'll have a short Q&A period and then a brief 5-minute break. We'll also have time for Q&A after other parts of the presentation. Please note, this webinar is being recorded. The slides for today's webinar can be downloaded from the Materials tab in the viewer. Please add any and all questions to the Q&A box in the viewer. Without further ado, I'll now turn the show over to Dr. Nazarov.
Thank you. Thank you, Janice. Thank you so much for having me here. Let me say a big thanks to the Antibody Society for sponsoring and supporting these webinars. And Janice, thank you so much for your guidance with the webinar. A big thanks to the Freestone platform for managing the technical side of webinars. And Julie, thank you so much for helping with setting up the webinar. And well, last but not least, thank you everyone for joining us today. This webinar was made with two goals in mind. First, I would like to give an introduction to Immunarch and quick start to help start using it now. And the second, I would like to give a direction for further research to help apply Immunarch, to help apply immunogenomics to immunotherapy with some plan in mind. I provide interesting publications along with the way, which will be useful to start thinking about immunogenomics analysis as a tool in a clinical setting, rather than just discovery or foundational research tool. It's extremely exciting to see the transformation of the domain, as I was in the domain since its inception more than a decade ago. And I think it's crucial to move from just, yet another omics technology to applied omics. The webinar and presentation slides will be available on demand for the future viewers, so please feel free to refer your colleagues here or rewatch it yourselves. Let me give you a very brief overview of my biography. I worked as a lead bioinformatician in a well-known laboratory of Dr. Chudakov's, the Laboratory of Adaptive Immunity. I worked on the intersection between T-cell biology and machine learning. There, I was a main developer of TCR, an open source R library for data analysis of immunogenomics data, and it is an Immunarch's predecessor. I worked on different indications. I work on ankylosing spondylitis, I worked on MRD, and I led a small but proud research group on machine learning in immune repertoires. After some time, I got invited to a role of director of AI in an artificial intelligence infrastructure startup, where I learned how to hire and manage team behind great products and how to build such great products with this team. And with all the new knowledge, I founded ImmunoMind when I returned to my first and true passion of T-cell biology. But I co-founded it with much higher skills and team and product management, in addition to science. I co-founded ImmunoMind with my friends and colleagues. We've known each other for a decade and worked together in the past. So today, the ImmunoMind team is the team behind Immunarch. That is an open source AIRR package. Combined with TCR, it has more than 200 citations and more than 45,000 downloads. Our team graduated from UC Berkeley Skydeck Startup Accelerator last year, and Immunarch is a very important a component of our multi-omics analytics platform for adoptive T-cell therapy development. In short, the platform helps select the best T-cell therapy candidates by comparing different designs with different cell population parameters, such as exhaustion, senescence, and other parameters. And Immunarch provides this important foundational immunogenomics analytics, in addition to single-cell transcriptomics on the platform. Last year, we raised our first investments, and we are working on partnerships. With Immunarch and our platform, we already helped several clinical centers and companies, and the resulting publications will be available soon. On this slide, you can see several fantastic publications that used Immunarch in immunotherapy. Please, after the webinar, feel free to go and search for them. Those are done by amazing researchers, and I highly recommend you to read them. And down below, you can see a couple of testimonials from happy users and amazing researchers with whom we worked in the past. And now let's dive into Immunarch. So Immunarch is a little bit different than other bioinformatics software. So to give you a reasoning on different user experience or bioinformatics decisions we made in Immunarch, I would like to share with you several principles we built Immunarch upon. The principles address typical challenges which arise from using bioinformatics software. So the first principle is that we would like to work on the first principle is the seamless work with data and methods. So there are many different data formats, and different research groups use different data formats, use different preprocessing tools, and it's important to support them all. And it is also important to support all popular or clinically important analysis methods. We follow this principle. The second principle is to minimize cognitive load to help focus in science. So the common story with bioinformatics software is when you have many tens and tens of different functions with complex argument names, with not so great help. So we decided to tone down the complexity of it and provide extremely simple to use, high-level tools to help people focus on science rather than programming. So in short, instead of remembering tens and tens of function names, you just need to remember 5. And instead of remembering names for different plotting procedures, you just need to remember only one, because in the code, Immunarch automatically detects what you're trying to do. The third principle is that Immunarch is data type agnostic. So we work with different data types in AIRR. We work with data frames, data tables, or databases if you have large scale data. And the interface, so the functions are still the same. So you don't need to learn another AIRR package to work with large scale data. No. The Immunarch is here for you. And the last one is that Immunarch is a data nature agnostic. So we support both popular data types. It's bulk sequencing and single cell sequencing. Let's move to the bioinformatics part. How to install Immunarch. First of all, consider using AIRR Studio for AIR data analysis. RStudio is an amazing software for AIR programming, and it's very helpful for beginners and advanced users as well. The most common installation method is to directly install the package from the CRAN by running this simple code line. In case of troubles, please feel free to check the Immunarch webpage for a very long, very comprehensive list of different solutions. Sometimes AIR is not very friendly in terms of package installation, but we invested a lot into finding different reasons for installation failures, and provided this list on the Immunarch website with, in addition to tutorials. And what's interesting is that if some of your other packages fail, please feel free to take a look at our list too, because sometimes it's a general problem rather than Immunarch specific or some package specific. The alternative to the CRAN installation is to install from GitHub. It is useful when you would like to install the latest development version. So, for example, when we develop a feature that is not in the CRAN release yet, you can just install the package from the GitHub by running these two lines. For this, you need to install the DevTools library. So it's like either two or three lines of code. And the last option to install Immunarch is useful when you work on servers. We provide what's called a Docker container. It's some sort of a virtual machine with all the pre-installed AIR packages required for Immunarch and Immunarch itself. In our Docker container, there is also an instance of RStudio. So if you know how to work with Docker, you can just simply install this Docker container and start analyzing data immediately without waiting for any installation procedures. Let's start with the data. So, as I already said, Immunarch supports all popular and critical T-cell and B-cell repertoire preprocessing and analysis formats. What's important here is that Immunarch automatically detects input formats without additional input from users. So the only thing that user needs to do is just pass the path to the file with immune repertoire or to a folder with a list. With immune repertoire files. On the screen, you can see currently supported formats by Immunarch. It's well-known Trust 4 or 10x Genomics or MyXCR formats. And of course, it's an AIR format support too. And we plan to implement some of the other file formats in the future, such as CATT. If you would like to request a support for a specific format, at the end of the presentation, I will share you with details how to contact us. It's all extremely simple. In order to load your data into R, you just need to run the function called repload. You provide a path to your folder or to your file and just wait for parsing. On the screen, you can see a screenshot with outputs from this function, and the function outputs the information on what files it automatically detected, and what files are not supported. In the bottom of this screenshot, you can see there is a file formats of RTCR and the other software that is not supported today, and function just skip them instead of failing with error. It's all extremely simple because we conducted quite a lot of interviews with researchers and realized that one of the hardest, one of the most painful part is to load your data into R for the subsequent analysis. So we decided to fix it. After loading your data, the data converts to the internal Immunarch data format that is extremely simple. Replot returns you a list with two elements. First element called data, and it's just a list of immune repertoire tables. In this immune repertoire tables, one row corresponds to one clonotype, and clonotype is a set of equal sequences with equal V-gen segments and G-gen segments. And the second element is meta. It's a table with metadata of immune repertoires. The data format, the data table looks like the following. You can see clonotypes in rows. You can see information about their proportion, about their count in the data. You can see their CDR3 nucleotide and amino acid sequences and other columns. The list of columns are in the right side of the slide. I hope it's clear. What's important here is that we also support additional columns, such as full sequence of the clonotype or other CDRs or a C gene segment name. It depends on the data you are working with. And metadata is just a simple table with additional information about repertoires. The left column is a column with sample names, and other columns they are some characteristics of repertoires. Again, I would like to remind you that on the Immunarch website, we have a very detailed tutorial on how to use it. So please feel free to check it as well. Let's start with the very first steps you should do after uploading your data. So exploratory data analysis. The goal of exploratory data analysis is to give you a very quick overview of your data for quality control and sanity check purposes. You would like to check if your data is okay, and if there are some anomalies in the data. There are different methods to do this in Immunarch. The most simple one, but probably the most effective, is simply a number of clones. So you can think about it as a sequencing depth of your immune repertoires. To compute and then visualize the number of clones, you just need to run the following lines. Take a look at the bottom left box. There are two different ways to run the analysis and visualize it. First, you can run the rep-explore function and then apply the function viz. And function viz is the universal function for any visualizations. So this function automatically detects what was the analysis on the previous step. In the current case, it's the rep-explore, and visualizes the results accordingly. Or you can use the little bit advanced syntaxes from the Plur packages, the Plur ecosystem packages, just run a simple line of code to execute and visualize the data. Here we can see on the left chart that data is approximately the same in terms of number of clones, so in terms of sequencing depth. So probably there are no anomalies, and it's fine. And on the other side, you can see an example of Immunarch's built-in grouping ability. If you have specific characteristics you would like to group by, such as immunotherapy response. So if you have samples with immunotherapy response and without it, or in this case, you have 3 different groups of patients with full response, it's the orange one, with partial response, it's the green one, and the group with no response, it's the yellow one. You can group by them. You can group the statistics by them and also see the statistical differences between these groups. Again, everything is done automatically in the viz function. This is not a great example of this statistical analysis, but it is much more important on later stages of the analysis. The second method for exploratory data analysis is the number of clonotypes, or in other words, it's the number of rows in your data. The idea here, again, is to find low quality samples or anomalies. So if you have some data with extremely low number of clonotypes, it's interesting, right? So you need to explore it, and you need to investigate it, whether it is some issues with an experiment, or it is biology, and it's quite interesting insight. Again, it's extremely simple. It's the exact same function of rep-explore. You just pass the volume parameter in the bottom boxes. So the first one, parameter in data, you just pass the list of immune repertoires to the function. And the second parameter is the volume, when you tell the function to compute the volume of immune repertoires or the number of clonotypes. Again, you can easily group the clonotypes by their status or by their response or by any other characteristics. The third feature for exploratory data analysis is the distribution of CDR3 lengths. The idea here, again, to find anomalies and differences between groups, and you can compute the CDR3 lengths either by nucleotide sequences of CDR3 or by amino acid sequences of CDR3. Here you can see how to do it in Immunarch. On the left side, again, instead of volume, you provide the length parameter and visualize it. And on the right side, you can see the grouped length distribution. On the x-axis is the distribution of CDR3 length, and on the y-axis is the number of clonotypes, or proportion if you pass additional parameters to this function. And the last method for exploratory data analysis in this small section is the distribution of abundances. So the idea here is to give a quick overview of the structure of a repertoire. It's a very rough estimation of diversity, about which we'll talk a little bit later. On the x-axis, you can see the different abundances. So the very left point is the clonotypes with only one clonal count. So it's a very rare clonotype. And on the right side, you can see the most abundant clonotypes. Of course, you can group these plots as well. The idea here is that you can look at the line. You can look at the skewness of line at the slope, and try to see the differences between groups or between repertoires. And again, what's important to remember here, that these functions just a rough estimation of much more precise methods, and they need to give you a quick overview of your data, to understand is there anything okay or are there some anomalies. This exploratory data analysis is what you need to do in the first place after you upload your data into Immunarch. And after you provide an exploratory, after you do an exploratory data analysis, you can go with downsampling. This is a highly requested feature because the goal of downsampling is to make data samples comparable. Some of the statistical methods simply doesn't work if you provide samples with vastly different volumes of data, with vastly different number of clonotypes. So it's very important to downsample it, to make immune repertoires almost the same in size, but artificially. On the left side, you can see the distribution of clones from the previous section of exploratory data analysis. So it's without downsampling. And on the right side, you can see the equal distribution. You can see the distribution of equal numbers of clones after downsampling the data to the least, to the smallest repertoire. The code for it is simply a ref sample applied to the imdata data object and with the method name downsample. You need to be a little bit careful here, because if your data is too small, then probably it is not a great idea to downsample all of your data to this smallest sample. Probably it is better to explore the reasons why it's so small and maybe filter out such samples from your data. So I would suggest always consider downsampling, but be cautious about these anomalies, that it's better to investigate them in parallel, without putting it in the data and skewing the whole analysis. And the last subsection in this small section is a well-known public clone type analysis. The idea here is to estimate the similarity of immune repertoires using the number of shared between samples clonotypes. Some of the applications include discovery of a tumor-specific clonotypes and prediction of response using very interesting indices. You can see the examples of using these indices in publications 2 and 3. And an additional application of the public clone type analysis is a preparation for further more advanced analysis. Because by running this pre-analysis, by understanding the connections, the relationships between your samples, you can make some hypotheses that you would like to test further. So again, in short, exploratory data analysis and downsampling and overlap analysis is what is better to do immediately after receiving the data, because it gives you the foundation for later more advanced analysis. In the overlap section, there are I provide an example of 3 different methods for public clone type analysis, but there are 2 or 3 times more in the Immunarch package. I just provide the most popular methods. The most simple and straightforward method is just to compute the number of shared clonotypes between samples. It works great if you downsampled the data beforehand, because in this case, you don't have these effects of different data sizes. Of course, in Immunarch, you can use CDR3 for comparison. You can use CDR3 nucleotide sequence or amino acid sequence, or you can compare. You can find shared clonotypes which have all gene segments, so V, J, and maybe even D, and CDR3 sequence as well. In Immunarch, we visualize it using the heat maps. On the left side, you can see default heat map visualization, and in the cells of the matrix, you can see the number of shared clonotypes. The right side visualization is a little bit more interesting because it provides an automatic clustering of samples, and that's why it is a foundation for further analysis. So in your data, when you apply the rep overlap function with the heat map 2 visualization, you want to look for these some sort of squares on the diagonal. If you look at the right side picture and at the bottom right side of this picture, you can see some sort of a red square consisting of other reddish squares. And this means that samples in this red square, so sorry, it's a little bit small for me. So samples S2, S1, S3, S4, S12, and S6, they are somehow similar in terms of shared clonotypes. And that's interesting. After that, you would like to apply some advanced methods for clustering of immune repertoires. I will talk about them at the end of the presentation. Or you would like to see whether this clustering is somehow related to your metadata. In order to find this red square, you need to look at the diagonal because this is a symmetric matrix. The next, so the shared clonotypes works greatly when you downsample your data, when there are some when the data is comparable, but it's still not a statistically correct way to do such type of similarity analysis. So what you want to do is to apply the Jacquard index. Jacquard index estimates the similarity of sample using the number of shared clonotypes, but also it normalizes by the data volumes. So you can so as an output you have metrics, you have numbers that tells you how similar different repertoires are. Because in shared repertoires you just have a number of shared repertoires, and you don't know what is the measure of similarity here. Jacquard index is a measure of similarity. What's important to keep in mind is that Jacquard index doesn't take into account the abundances of clonotypes. So it just looks at them at all equal. This will be important on the slide after the next one. To visualize Jacquard index, you need to run the rep overlap function with the Jacquard method name, bottom left box, and apply the one function that rules them all, the viz function. As you can see, there are no numbers, there are no shared clonotypes in the matrix cells, but numbers which demonstrates you the similarity of different repertoires. And again, the second visualization with heat map 2 provides more interesting analysis here. Okay, but maybe you also would like to include the abundances in the analysis. So you would like to not only compare the sequences, you also think that in your analysis it is important to include abundances because of the immune response. So with the immune response, if two samples have the same immune response, they will have, well, probably they will have the most abundant clonotypes. And to measure that, you can use the Morishita-Horn index. It's just like the Jacquard index, but it also uses the information about abundance, about the numbers of clonotypes, the numbers of rows in the tables, in repertoire tables. And what's interesting that it heavily affects the result. And as you can see, there is simply no clusters in this type of analysis. So just to remind you about Jacquard and shared clonotypes, who found some sort of cluster, right? Of course, it's not a cluster, but it's some sort of cluster. When Morishita-Horn index didn't find anything except this very strange anomaly of two samples. And that means that this cluster we discussed on the previous slides, the clonotype, the repertoires, they're similar in terms of overall clonotypes, but they are not similar. They're not similar in terms of distribution, in terms of abundance of these clonotypes. It's important to consider. And again, it depends on your hypothesis behind your data analysis. The next section will be about clonality and diversity analysis. I think it's probably the most interesting section in immunotherapy because it has the widest application to immunotherapy. So maybe we can answer some questions. 1 second.
Okay, I'm back.
Oh, thank you.
Takes me a little bit to find our spots to click on. Questions. We do have a few. First question is, how does one merge technical repeats?
It depends. Again, it depends on the type of your analysis and what would you like to do with it. So I will not talk about this merging in the presentation, but there are different ways to do that. For example, if you do it for improving the precision, right? You can merge it with you can merge the samples and apply some function to abundance, maybe mean, maybe median, to the clonotype counts. However, maybe it's more interesting to use replicas for bootstrapping procedures. So again, I will not talk about this in these presentation slides because it's very advanced. But maybe you can use replicas to increase the accuracy of clonality, diversity, and other measures by integrating replicas into the bootstrapping procedure that runs clonality and diversity measures on different subsamples, right, of your data. I understand that it's a little bit high-level answer, right? But I hope I helped here.
Thank you. Next question. What does downsampling do to singletons? Just sample random ones, not considering probability of generation.
Right, thank you. So in the rep sample function, again, just a reminder, we have a tutorial on it on our website. It is possible to use different methods for downsampling. So the current version, the rep sample with the downsample, it's the simplest one, right? And it works great. It's just randomly, it doesn't use any probabilities. It just randomly takes, it's like in a statistical exercise, right? It randomly takes spheres of different colors from a bag with these spheres. So singletons, some of the singletons will get to the resulting immune repertoire, and some of them don't. We have different sampling procedures, including probabilities, including the one using probabilities, and you can read about them on our website, or maybe in one of the next webinars.
We had a quick refinement of the original question about emerging technical repeats, and this is what was meant was more if it's possible in red load or if it must be done manually.
Yeah. So not yet. Yes, if you have some suggestions, if you have best practices on how to do it, please contact me. Our team will be happy to implement these best practices to help researchers.
Next question, would it be possible to open FASTA or FASTQ files with Immunarch?
Thank you. No, because Immunarch is focused on post-analysis of immune repertoire data. So on the first step, you apply what's called CDR3 extraction tools, such as MyXR, IMGT, 10x Genomics, and so on. And then you supply the output of the CDR extraction tools to Immunarch. So there are two large steps, and we focus on the second one specifically.
Thanks very much. And I will just read the first part of this because it is, hi, very useful presentation. Thank you very much. Okay. And then the question is, can you compare your platform in comparison to IMGT HiQuest? Is it and is it available for academics?
Right. I think there is a little bit misunderstanding. Sorry if I didn't make myself clear. The Immunarch library is open source, completely free to anyone. It's available for academia. It's available for industry. You can use it however you want, right? Our ImmunoMind's adoptive T-cell therapy platform is proprietary, and it's a topic for, if you're interested in it, please write me an email, but it is not a topic of today's webinar. So, and regarding the IMGT High request, IMGT High request returns the processed IMGT High request is the CDR3 extraction software. And there are, if I remember correctly, there are some basic statistics, but it seems like it is not enough for widespread analysis. So the typical scenario I see in publications is when people ask for, people use IMGT High request for extracting of CDR3 sequences, and then use other software like Immunarch to analyze the data.
Thank you very much. Well, that concludes the questions for this section. So we're going to take a very brief break here, about 5 minutes. So please convene, come back. We'll be here at 11:55. See you in a few minutes.
Thank you. Mhm. Let's continue with the most interesting section that is about clonality and diversity. Just a quick introduction to this topic. So clonality analysis and diversity analysis, they are extremely similar. Their goal is to assess the diversity of immune repertoires from a little bit different perspectives. Clonality analysis pays more attention to anomalies in the abundance of clonotypes. So, for example, if you have if a patient has a cancer clonotype, it will be extremely overabundant, and the clonality analysis will pick it up. The diversity comes from the ecological and microbiome studies, where researchers are required to count the number of unique species in the environment. So there are different to clonality methods. They are focused more on the estimation the true number of clonotypes in immune repertoires in samples, right? And clonality and diversity, they are starting to get more and more attention as biomarkers for therapy response prediction. The reason for it, I think there are several reasons for it. The first one is that diverse T-cell receptor or immune receptor repertoires simply has a greater probability of containing very or rare clonotypes, which might expand into dominant clonotypes later to support, again, the late but robust tumor response. The second reason is that multiple different receptors can recognize the same antigen epitope. So, simply put, the more receptors you have in your the more different receptors you have in the immune repertoire, the higher the probability that they will magnify the immune response to an antigen by simply creating this pool of diverse T cells or other cells that attacks that recognizes the same antigen. And also there are some studies, again, recent studies that indicate that immune receptor repertoires, they are affected by immunotherapies. So probably you can catch some adverse effects before the manifestation of these adverse effects just by assessing the diversity. So let's start with the clonality, and I again highly recommend you to download the slide deck after the webinar and go to links in the highlighted publication sections in every analytical section here. So clonality analysis. Clonality analysis, the goal here is to estimate and, of course, compare the differences in abundances of immune receptors between different samples. There are several methods to do it. The first method is the proportion of the most abundant clonotypes. So as I said, the idea of clonality is to look at these anomalies. So let's look at any interesting anomalies or any interesting distributions among the most abundant clonotypes. This is done by first sorting the immune repertoire by the immune receptors by their abundance, and then enumerating them from one to the last one, starting from the most abundant clonotype. So I know it's a little bit sophisticated, but let's look at the chart, at the left chart. The legend says the clonotype indices. The first one, the red one, 110, means that this pool of clonotypes contains first 10 clonotypes. The second pool of clonotypes, orange one, contains clonotypes with indices from 11 to 100, and so on. Then we just combine the abundance of these clonotypes and plot it on the per sample chart. So if you look at the very first column at the left for the S1 sample, you can see that the red pool, the abundance, the summary abundance of the first 10 clonotypes, it is around, I don't know, 10%. So it means that the first 10 clonotypes occupy around 10% of the overall immune repertoire in the sample, for the sample one. It's not clear, is it good, is it bad, because it's important to have a hypothesis about the immune cell biology in mind, and to compare against it and against different samples. So, for example, the second column, the S10 sample, it has much less abundance in the 10 most abundant clonotypes. That's interesting, right? So in order to compare it, again, Immunarch provides the grouping ability, and this is the function you already saw. It's the viz function. The function to analyze the clonality is the rep clonality, so just repertoire clonality. And then you just pass the result to the viz function with the very typical, for this presentation, grouping parameters. So let's group them by response: full response, no response, or partial response, and group it by a metadata table we have. And here we don't see any critical anomalies. But what's interesting is the second group of columns from the left, from 11 to 100, is that the full response group has the lowest abundance for these clonotypes. The green group is a partial response, right? Has the average abundance, and the yellow group, no response, has the most abundance. So it means that in the group of patients with no response to the immunotherapy, the first 100 except first 10 clonotypes occupy more space, so they are more abundant than in other immunotherapy patients. After that, you may want to look into it. You may want to find what are those clonotypes. You may look on the first, I don't know, 50 or 20 or 30 clonotypes to see whether they are interesting, whether they are associated with cancer, with the specific cancer, whether they know about them from your previous research, and so on. The second approach is the opposite to the most abundant clonotypes. It's to look at the least abundant clonotypes. The idea, again, the same, just we just look at the least abundant clonotypes and do the counting a little bit different. Here, for bioinformaticians, here we just compute the weight of the tails, because immune repertoire receptor abundance distribution is approximately zip distribution, and it has quite a heavy tail. The idea here is simple. Let's pool clonotypes not by indices, but by their clonotype counts. So let's pull together all the clonotypes which has only one count. So you encounter them in the sample only once, and sum up their abundance and plot it on the plot, and do the same for other clonotypes as well. So let's put together clonotypes with 2 or 3 counts, and so on. And here you can see, you can look at anomalies in these very heavy tails, in this distribution of very extremely rare clonotypes. It seems that it's interesting to see, again, it's interesting to see whether there are too much tail or whether there are not too much. This is the an opposite to the previous method, so I won't tell about it anymore. The last method in the clonality analysis is what's called relative abundance. So it's some sort of, it lies in between the previous two. It's the most complicated, but the most powerful one. The idea here is to pardon me. The idea here is to split the repertoire to different groups, basing on their percentage or proportion in the immune repertoire, and then sum up these clonotype proportions and plot it on the plot. So how it looks? Let's look at the left side, at the left chart, and let's look at the legend. There are 4 clonotype groups. The small group consists of clonotypes which occupy, you see, from 0, right, to very small, to extremely small number, 00015. The second group, let's call them medium clonotypes. They occupy the second set of abundances, and so on. So if you have some idea on how clonotypes in your domain, how clonotype abundances in your domain look like, you can create these groups and look at the specific group you are interested in. So, for example, if hyperexpanded groups in your domain is something that occupies more than 10% of repertoire, you just pass this information to the rep-clonality group with the Homeo method name, and you can easily compare it between your samples, as you can see on the right side. So the right side, I know it's a little bit different to the left side, right? But the x-axis is these clonotype groups, and the y-axis is the proportion of these groups in different samples. That's why you see small dots on top of bars. Every dot is a sample. Let's move to the diversity analysis section. After that, there will be one more very small section about gene usage segment analysis, and I will answer your questions about this section. So please feel free to write your questions about the clonality now. The diversity analysis is more complex, and its goal is to estimate the true number of clonotypes in immune repertoires and, of course, compare it. The true number of clonotypes is a quite complex task to estimate, but it is possible to give rough estimations that is useful in clinical settings. So diversity analysis used in patient selection, used in response prediction, and used in prognosis biomarkers. Analysis that is quite interesting. Again, take a look at these publications. They are very interesting. There are some of them about CAR-T cells. And the first method of the diversity analysis is the well-known Chow-1 index that is heavily used in the ecological settings to estimate the number of species. In our case, it's clonotypes in immune repertoires. It's one of the most, if not most, popular method for data analysis of immune repertoires in general. Of course, we provide it. The function to do it is the repertoire diversity function, or rep diversity. And you just pass the Chow-1 method name to it. On the left side, you can see different Chow-1 indices, results for samples, for different samples. And on the right side, you can see the same samples grouped by their response. And as you can see, the orange group of full response has the most diversity. The green group of partial response is somewhere in between, and the yellow group of no response has the least diversity. This is a very typical picture in immunotherapies, and it may be extremely useful for you if you don't use it yet. So it's a great biomarker, and it I encounter the diversity as a biomarker quite frequently. A small comment on the true number of species or true number of clonotypes. Of course, it is not. It seems that the Chow-1 index, despite being extremely useful in ecological studies, it doesn't give the correct answer for immune repertoires. And there are very interesting publications with more advanced diversity indices which show that it is possible to do more precise diversity estimations. But what's important that before the research community comes up with the more advanced and very precise diversity measure, the Chow-1 index is a simply amazing tool for measuring the diversity and linking it with clinical outcomes. So please consider using it. The second type of analysis is the rarefaction analysis. Again, it has its roots in the ecological studies. The idea here is the same: as Chow-1 is to estimate the number of clonotypes, estimate the number of true clonotypes, true number of clonotypes in immune repertoires. But the technique here is a little bit different. The rarefaction analysis extrapolates the data. It models the situation when there are more data in the samples. It looks like this. Let's take a look at the left side. At the left side, you can see the dotted lines, which are extrapolations of the samples, and at the around 0, you can see straight lines, which are interpolations. So it's a true data, and dot lines are the modeled data. These refraction curves provide a little bit more information in contrast to Chow-1 because of the lines, because you can see the behavior of these lines. And it also works in samples with when you have some troubles with sequencing depth, or for example, samples with vastly different sequencing depths. Again, it's better to filter out bad samples. But if it's hard to do downsample, probably you can do some sort of small downsample. So not to downsample to the smallest immune repertoire, but downsample to the smallest times 3 immune repertoire. So if your smallest immune repertoire has 100 clonotypes, downsample others to 300 clonotypes, and then apply the rarefaction analysis. But still, this looks more like a workaround rather than solution. So please be careful. Please be careful with it. And so in the function, the rarefaction is almost the same as Chow-1, and it is possible to group these lines, these samples, to show differences between groups. And again, you can see that full response group is the most diverse one, a no response group, the yellow one, is the least diverse one, and the partial response group is somewhere in between. A little bit on the more complex side, there is a method called Hill numbers. So I think it was pioneered by Dr. Victor Grief, where he applied the machine learning to Hill numbers to classify different mice repertoires. The idea here is to compute traditional diversity indices with different parameters and build a line of this, build a line of the number of clonotypes depending of these parameters. So there are some nice explanations on the web. We have a small tutorial on our website about it, so I won't go into much details, but the idea here is that the idea here is that different q-values, this is a parameter that you set up for the diversity computations. The different q-values, they give different weights to different clonotype abundances. So by trying different q-values, you can have some sort of an overview of the overall structure of the repertoire. It's a little bit similar to the relative abundance we discussed in the clonality section, but it's similar in terms of the idea that you can have an overview of the repertoire. I didn't encounter this method in the immunotherapy publications, but I think it's interesting. I think it's interesting specifically because of giving this structure of the repertoire, and in other words, an additional information about repertoires rather than just the estimation of the true number of clonotypes. And the last subsection in this section, after which I will be happily answering your questions, is the gene usage analysis. As we know, immune receptors consist of frameworks, consist of CDRs, and of different gene segments called variable gene segments, diverse gene segments, and joining gene segments. They occupy a lot of sequence space of immune receptors, and it was shown that it is possible to link them to different disease to different indications, so they probably can work as a patient selection in application of patient selection, and it is possible to use them for response prediction and prognosis, immunotherapy response prediction and prognosis biomarkers. Please feel free to take a look at this publication. The first one is about adoptive T-cell therapy, where these fantastic researchers used gene usage, and the second one is about ankylosing spondylitis, is about autoimmune disease and associations between patients with this disease and specific gene segments. It looks in the Immunarch, it looks like this. It's all gene segments for TCR beta chain, so that's why it's a little bit small. The call to the function is, as always, extremely simple. So it's just gene usage, gene segment usage applied to the imdata data object and visualized with the one and only viz function. And of course, you can easily group it by different statistics. So here we group it by response, after which you can explore whether there are some interesting gene segments that are different between groups. So on this plot, on the very top of the plot, you can see many ones and 0.77. It is p-values for Kruskal test between groups inside a specific VGen segment. So again, Immunarch does it for you automatically. You just call gene usage and then visualize. As this is the same statistical tests that are being applied in other grouped visualizations as well. We have one more section about the annotation and tracking. Again, I think extremely interesting and quite undervalued, I would say, methods for data analysis for applying the immunogenomics in clinical settings. Right. So the first question is about slide 44. Yeah. So of course there is. Thank you so much for this clarification. Of course, the 5 here is not the the true number of samples. I think we need to improve the text. The text here is just order of the clonotypes, and the same for x-axis. Thank you so much. And the second question is about slide 35. Do you know how many cells were sequenced in this example of immunotherapy? No, sadly, I don't have this information. Yeah. any more questions? Okay, so let me continue with the annotation and tracking. And I will leave my contacts at the end of the presentation to, one second. So the question is, can the same methods be applied to B-cell receptor sequencing? Any questions regarding analyzing B-cell data? Yes, of course. Of course, I just have examples about T-cell receptor repertoires, and I just provide you these foundational methods for analysis of any immune receptor repertoire data. The general caution here is that be careful with the applying. So T-cell receptors and B-cell receptors distributions of abundances, they are a little bit different, to say the least. So you may, if you apply the same methods to B-cell receptor analysis, the pictures here may be completely different. And that's totally fine, because T-cell receptor data and B-cell receptor data are completely different types of data. In case of B-cell receptor data, we have these somatic hypermutations. So the diversity simply by default is much larger here. Thank you for the question. I think this is an amazing question.
Yeah, I apologize. I disappeared there for a moment. I somehow got locked out. Thank you for jumping in there and soldiering on.
Right. So I don't see any questions yet. So let me continue with the annotation and tracking. The annotation and clonotype annotation, clonotype tracking, they're extremely similar. The idea here is to characterize clonotypes in your immune repertoires using either external databases, and that's in this case we call it annotation, or internal databases developed by you. In this case we call it tracking. And these databases contain any useful clonotype information, such as links to specific diseases. I think it's an extremely important method, right, or family of methods or approach to consider, because with the annotation you can characterize your immune repertoires. You can see what is the. So suppose you analyze the data, and if you take the most abundant clonotypes, you can find whether they are linked to a specific disease or not. Maybe it is important if you do some infectious disease analysis when you look at different time points. There are some information about immune receptors links to specific cancers. Specific cancers. Maybe it will be useful for you. And the tracking is when you have this database but built internally. So if you have already known list of clonotypes you would like to look at, for example, in case of CAR T-cell therapy, you know the list of clonotypes you would like to follow, and with using tracking you can assess the persistence and expansion of CAR T cells across different time points, across different patients. Here I provide a couple of publications. So it's again, as always, it's extremely interesting. Please go ahead and take a look at them. The the last publication is extremely interesting because it is done by Juno Therapeutics, and they apply both single-cell transcriptomics and immunogenomics to data analysis and track cells after infusion. So if you do CAR T, if you do any other T cell receptor therapy, adoptive T cell therapy such as T cell receptor therapy, please consider annotation and tracking. It is possible to do it, and it's very useful. So in case of annotation, you just want to link clonotypes in the sample to the specific diseases, to the specific conditions, using external databases, and maybe compute some statistics on top to see whether there are some interesting insights. So let's consider this example in which we order. It's a little bit different data. In this example, we order the samples by their time points, and you can see the development of different clonotypes, how they grow across from time point to time point. To loading. Oh, and sorry, I forgot. Immunarch supports all modern databases with clonotype information. So it's VGJDB, MacPass TCR, and PyRDB, PyIRD database. The R syntaxes for loading databases is a little bit verbose, but you just need to load the database one time with the function called dbload. In this function, we provide a path to the database file that you download from the database website. You provide an identifier for a database, and you provide a species, you provide chain, and if you would like, you can also provide the indication. And then you just call the function dbannotate and visualize it. So the CDR3-RR and CDR3 beta amino acid here is just additional. There are parameters you pass to the function to link together data of the same type. So here we want to annotate the immune repertoires using CDR3 amino acid sequences. So you pass the CDR3 column and to indicate that you would like to use amino acid sequences, and you in your data, and you pass the CDR3 beta amino acid name to the sequence to the function as well, to indicate that you would like to choose this column from the database. So if you would like to link together more than one columns, you can also do it in the exact same way. You just provide more columns. And tracking is the same as annotation, but you can track your own internal database. So for example, if you develop specific T-cell receptor, you would like to see how it develops in different patients and different time points. So you create some sort of your own database. It's just a list. In case of Immunarch, you just need to provide a list of sequences and track it with the track clonotypes function. For the track clonotypes, you provide the imdata object. So the list of immune repertoires, you provide the target. The target is simply a list or a vector of sequences you would like to follow. You provide the column that we would like to follow, the amino acid column, CDR3 amino acid column, and visualize it with the most popular function in Immunarch, the viz function. If you would like more flexibility, it is important. It is possible to rearrange time points. If you can see that we pass the additional parameter called sample order to the with function to indicate in what order we would like to see the sample. So if you have an additional metadata stored in the imdata meta about the time points, about the time from infusion or something like that, you can use it to rearrange the samples with one line and plot these nice plots. And if you would like to do some quick and dirty analysis of some of your immune repertoires, you can track the most abundant clonotypes from a specific repertoire. So here we pass the list with 2 elements, 1 and 10, to the track clonotypes. The idea here is to take the first 10, the 10 most abundant clonotypes from the first sample and track it across all other samples. As you can see, there is not so much success here. We found the samples only. The second one is in the S10. So please don't panic if you see such chart. It's okay. You just don't have such clonotypes. We are on the very last slides here, so probably it will be better to answer your questions about tracking annotations if you have any. And I will go to my last slides to talk about advanced topics and future of Immunarch. Let's give it 7 more seconds, and then I will go to I will continue with my last slides. Okay, so let's continue with slides. I didn't cover some topics, quite a lot of topics. So just a reminder, the website immunarch.com contains tutorials for all topics I discussed today and some of the advanced topics. There is a button called Start Here on the Immunarch website. So you just click on it and you will see all the tutorials. There is a cluster, a family of methods I named data-specific analytics. So first of all, it's B-cell receptor lineage trees analytics. We will release this functionality in November this year, but we already have a development version. So if you'd like to test it, to try it, and to drive the development, to maybe to suggest something you wanted to do for a long time, please feel free to contact me or open an issue on the GitHub. I'll talk about it on the next slides, how to do it. We will be happy to work together. Single-cell immunogenomics, in other words, the paired chain data. So the data I showed you earlier is single chain data. So there is either TCR alpha sequence information or TCR beta sequence information. Immunarch also supports paired chain data. So 10x we support parsing the 10x genomics paired chain data format, and the basic support such as diversity tracking on overlap computations or exploratory data analysis is working in Immunarch. Of course, we'll be working on improvements, so any suggestions are welcome. If you work with transcriptomics and immunomics data and you work with them, well, you would like to somehow integrate it, please feel free to contact me. and single-cell transcriptomics, site-seq, and other very exciting single-cell data types. We didn't support it in Immunarch. We have support for it on our analytics platform. But not supported in Immunarch. But we plan to someday open source our internal software tools, just like with Immunarch. The second cluster of topics I would like to discuss is the advanced statistical analysis. So first of all, it's possible to do advanced gene usage when you apply linear models to gene segments and identify specific gene segments linked to a therapy response or something like that. There are some advanced diversity indices I already mentioned before. They are computational heavy, and it's important to again be a little bit careful here because the methods are complex, so you need to clearly understand the assumptions they are built on. There are no such advanced diversity indices in Immunarch yet, but we are looking on it and looking for a gold standard method that we could either simply implement or optimize and implement. There is a whole section regarding the post-analysis. So, for example, when you compute overlaps, and as an output, you give this matrix of similarities between immune repertoires by their shared clonotype numbers. The same can be done with gene usages when you compare distances between gene usages of different immune repertoires. As an output, you get exactly the same similarity matrix. It is possible to do some interesting analysis on top of these matrices, such as Cummings clustering or PCA or other complex advanced statistical analysis methods. Some of them are supported in the Immunarch, such as clustering and PCA. There is a whole topic on public repertoire analysis. So instead of simply computing the number of shared clonotypes, it is possible to create a very, very large table of all clonotypes and work with it. So it is possible to create a large table with clonotypes with an external database and compute some interesting statistics such as distributions of abundances for disease-specific clones in different repertoires. So you can compare different groups, different repertoires, or different groups by the abundance of cancer-specific or CMV-specific clonotypes taken from database. It may be useful for classification. One of the publications in the annotation sections is about exactly the same. And there is a CAMER and sequence motif analysis. The CAMER analysis is supported in Immunarch, and sequence motif analysis, there are quite many fantastic tools for computing sequence motifs. This is the area that is being heavily developed in the last 2 years, I think. So we are a little bit hesitant to implement or to integrate any of methods because we would like to wait and see whether it is possible to provide this quality of analysis we strive for, or is it too risky. But again, if you have any suggestions or ideas, please feel free to message me. And a couple of words a couple of words regarding the improvements. We plan to improve the user experience. So, for example, we plan to post more verbose error messages to explain what went wrong and how to fix it. We plan to do more convenient visualizations. So, for example, in the current version, you need to pass the meta object to charts, and we have plans how to remove it, and you just pass the by, so this by response, and your data is grouped. And of course, we plan to do more tutorials and learning materials, so feel free to check our websites and socials for more tutorials on Immunarch and some info on CAR-T. How to communicate with us? The most preferred way is, of course, GitHub issues, because it's a centralized place for development, and the communication is extremely convenient. Here is a link to the GitHub issues. So if you have a question, a suggestion, a bug, you just need to register on GitHub and open a ticket here. But if you have some sensitive information, if you would like to share or discuss some sensitive information, we have an email support on ImmunoMind.io. We don't check it very regularly, I'll be honest with you, because we are sitting on GitHub in GitHub issues, but we do check it. So, and here's my contacts. Please feel free to contact me about your projects, about your T-cell therapies or CAR T-cell therapies. I'll be happy to discuss them. Please feel free to add me on LinkedIn and write me an email or write me a message on LinkedIn. On the right side, you can see the link to our website on Immunarch and the direct link to the GitHub code of Immunarch. Thank you.
Thank you very much. We just have one question, having already very efficiently dealt with most of the ones that came in. Actually, nope, we've got another one. I lied already. Okay, first question. How do you proceed for rarefaction analysis for paired chains with 10x or Chow1?
Right. So currently, it's the most straightforward method. We just merge sequences to get this super clonotype with the pair chain data and compute it. So there are some challenges with this, and I think the overall challenge with the single cell support, with the support of single cell immunogenomics, is the very definition of clonotype. Like, what is the clonotype in paired chain data? Because there are some different approaches to do that. So in order to minimize any risks and make it more convenient, it is possible to load 10x genomics as a separate pair chain files to compute diversity pair chain. I hope this answers your question.
Thank you very much. We have one last question, unless somebody makes a liar of me again, and that is, what is the size limitation or number of sequences?
There are two variables. The first one is R limitations, and the second one is limitations of your server or personal computer. So probably if you have more than several hundred of sequences per repertoire, it is better to use the data table format rather than data frame format. the data frame format is the default table format in the R language, and data table is a simply amazing package, highly optimized package for working with large scale data, and probably it's better to move to it. To be completely honest, I would highly recommend anyone to learn the data table package and simply work with data tables 100% of their time, because it's much more convenient and much more faster. But we also can work not everywhere, but in most of the functions we can work with databases. And also we have so database is a semi-experimental support, and we have a more experimental support for Apache Spark data frames. In case you have, I don't know, 1 million clonotypes per sample, right?
All right. Well, the last comment from the attendees is, thanks a lot for this webinar. Bravo. So in concluding, I want to thank Dr. Nazarov for relating his insights into easy, fast, and practical error analysis. An on-demand version will be available in a few days. I'll send a link to all the people who registered, and you'll get this by email. Please do feel free then to watch this or any of our on-demand webinars when it's convenient for you. Thanks again, and have a great rest of your day.
Thank you so much.